Hello, everyone, and welcome to today's webinar, From Bench to Bedside, Targeted Gene Editing. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Millipore Sigma. To learn more, visit sigmaaldrich.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Jennifer Bennett, PhD, Product Manager, Millipore Sigma. Jennifer, you may now begin your presentation.
Thank you for that introduction. Hi, all. As she said, I'm Jennifer Bennett, and I'm excited to tell you today about some of the products that we have from bench to bedside for targeted gene editing. First, the life science business of Merck KGaA, Darmstadt, Germany, operates as Millipore Sigma in the US and Canada. Creating therapeutics has been the purview of large pharmaceutical companies who have the budgets and the production equipment for lengthy discovery, preclinical, and clinical studies. But as therapies have expanded from small molecules to include novel modalities like monoclonal antibodies, antibody drug conjugates, and cell and gene therapy, these have expanded out in the number of companies that can work on them outside of pharma companies. In parallel, the tools to innovate and generate these therapies have expanded, becoming less expensive and more accessible to researchers outside of the pharmaceutical companies. However, all researchers still need to follow this high-level paradigm. First, designing the experiment. What are you trying to solve, and how do you plan to do it? Evaluating and validating. Choosing and validating your reagents. Of course, then creating the model that you're going to use to both test and build your therapy. Evaluating or assessing your model by whole model approaches, including high content imaging, genomics, proteomics, metabolomics sometimes, whole genome sequencing, and then redoing this multiple times during the discovery process until this is optimized for efficiency, where then we move into the translation phase, where the model can be tested for safety and efficacy in the clinical trials. And we provide the tools and expert technical support along this workflow for every step through this. And so we're going to focus on an application, targeted gene editing, that mirrors this workflow today. So functional genomics sits at the nexus of discovery using sequencing and bioinformatic tools and applications in understanding and treating disease. Functional genomic tools help us to evaluate the complex junctions between genotype and phenotype and often help us move from theory to practical application. Functional genomics is a practical science designed to bring together the genomics and the phenotype closer together and connect all of these as you move toward your therapy. And the revolution of engineered models using functional genomic approaches helps to identify phenotypes, isolate critical factors, determine biological mechanisms, assay outcomes, assay new novel drugs and modalities, create experimental models in both animals and cell lines, and of course in bioproduction. One area of functional genomics that has grown, of course, in the genome engineering over the years has evolved toward more precision in gene modification and editing. New discoveries have been recognized and created more efficient engineering tools, from chemicals that could create advantageous gene mutation to short stretches of RNA that could silence genes, to proteins that could be engineered to both bind and cut specific sequences of DNA. These have continued to evolve, and the most recent of that is CRISPR. CRISPR, in fact, began as a study of the bacterial immune system, but quickly became a tool for creating model systems and investigating diseases, specifically those that could not be investigated from previous gene editing systems. CRISPR screening technology opened new doors by providing versatile and simple tools for high-throughput investigations. And CRISPR has moved very quickly into the clinic. In just a decade, 5 or 6, in fact, new therapies have been approved by the FDA, and over 50 clinical trials are currently being conducted using CRISPR gene editing reagents. But CRISPR continues to evolve as new variants and new combinations are invented to make that editing more precise and safe for clinical use. So when people talk about the uses of CRISPR and CRISPR-mediated gene editing, they're usually talking about nuclease-based creation of double-strand breaks. When using nuclease-based CRISPR technologies, we're really relying on endogenous DNA repair mechanisms to achieve the desired modification. The outcomes are achieved either through non-homology end joining or homology-dependent repair, or HDR. There are some tools and strategies that can shift this balance a little bit between these pathways, but it's not really your choice which mechanism the cell uses to repair the double-strand break, and it predominantly uses non-homology end joining, often resulting in loss of function mutations caused by frame shifting, insertions, or deletions. One important difference between these two pathways is the error-free repair and error-prone repair compared to repairing off of the plasmid. Homology-dependent repair requires the addition of a donor DNA template for which to pull the DNA off and integrate into your target gene of choice. Of course, when this repair pathway is done endogenously in the cell, it often uses very similar sequences off of a sister chromatid, but in this case, you'd be introducing the DNA of interest that you'd like to integrate. Again, the default pathway tends toward non-homology end joining, and HR is mostly inefficient in many therapeutic cell lines, which makes this an area of interest for increasing this to get new mutations and substitutions. With CRISPR and with other genome engineering tools, you can achieve a wide array of genetic modifications. So here we're looking at various places on a gene where we can add in, make modifications using genome engineering tools, specifically with CRISPR as well. The predominant application, most common, is usually a gene knockout. And again, this uses that non-homology end joining where creating a double-strand break in an exon results in insertions or deletions of nucleotides, leading to a frame shift and incorrect expression of that gene, generally a knockout. But there are multiple other genomic manipulations that can be achieved. Again, we talked about with HR integration. So for example, fusing a reporter gene, putting in a GFP construct behind your gene so that you can follow it in the cell line. This can be done with CRISPR by putting in a donor GFP sequence to be integrated at this site. The choice of which type of genetic modification is critical to the success of either the model that you're creating or the therapeutic you're designing. So let's take a look at how some researchers have used CRISPR in their drug development pipeline. PDE10A is a phosphodiesterase that inactivates both CAMP and CGMP and is almost exclusively expressed by neurons in the striatum, a brain area often targeted by antipsychotic drugs. Researchers sought to inhibit PDE10A as a novel therapeutic avenue to discover antipsychotics. They used the fluorescently tagged triple knock-in U2OS cell line, featuring endogenous tagging of 3 gene targets with different fluorescent proteins. So we have alpha-tubulin tagged with GFP in green, lamin B1 is tagged with BFP, and beta-actin is tagged with RFP in red. A cell-based assay involving high-content imaging was used by researchers to identify a microtubule aggregation phenotype that, along with other phenotypes, was used as a measure of toxicity. In A, we have a DMSO control when added to the cells that have been tagged. The cortisol is a known is a positive control. It's a drug known to create microtubule aggregation, and that's seen here in those pictures. The researchers then tested two different compounds, compound 8148 and compound 0558, and when added to the cells, they could tell the difference in the phenotypes by the tagged microtubulin. From these assays, researchers were able to eliminate compounds that would have resulted in late-stage failures, costing the company millions. CRISPR is enhancing the early discovery workflows to provide more accurate information before moving into those preclinical stages. Often, discovery work will include functional genomic screening at the whole genome, pathway, or family level to identify crucial targets for action. And as we move down this funnel, the gene editing workflow decreases in scale while increasing in specificity and efficiency. While screening is an important tool for which we offer several off-the-shelf and custom reagents, in this webinar, we are going to focus on editing of single genes. So whether you're starting from functional genomic screening hits that have been validated, or you know your target of interest already and want to just attack it without having to go through screening, this next workflow can outline is a very basic outline for how to perform these CRISPR experiments. So again, the first step is to determine the type of modification you need to make and in what model system, and it will have a large impact on the downstream design. Next, we need to design the guide RNAs and choose a nuclease. This step can actually be quite overwhelming for new users. There's many different varieties of nucleases, many programs with which to design guide RNAs, and many places to target those guide RNAs. Using our CRISPR design tool, you can actually put in the gene ID or a custom sequence and get back guide RNA designs that we've stratified by efficiency and specificity for you. We also have pre-designed guide RNA sequences targeting the majority of the mouse and human genomes that have been validated, and they come from different libraries like the Sanger library. Then it's a matter of choosing the appropriate nuclease. For example, will you be using a wild-type Cas9, our new Cas9 Plus, a Cas9 nickase, and which format are you going to use? Nucleases and guide RNAs can be expressed via plasmids, mRNA, lentiviral constructs, or as purified and synthetic material. Each format has its limitations and its advantages depending on your end goal. At this stage, it's important to clarify your end goal to make sure you're using the appropriate reagents. Do you intend to use CRISPR reagents to modify a cell line that can be used in cell therapies, or are you making a disease model line that you can use to screen drug compounds? Those are just two examples of the many uses, but knowing this up front and selecting the right reagents will save you time and trouble down the line. The next step is to introduce your CRISPR complexes into your model system of choice and evaluate the modification efficiency. Ideally, you would choose several guide RNA designs and introduce all of them in parallel to choose the ones with the highest efficiency. A T71 digestion can give you a quick evaluation to eliminate the guide RNAs that are not going to work. And further sequencing, protein expression, phenotypic or morphological assays can be done to confirm the guide RNA is in fact creating the target or creating the modification that you want. Additionally, off-target analysis can be done at this stage. Specificity for the target sequence is a key component of every CRISPR complex, and the more you know about possibly deleterious mutations outside of your target gene, the better suited you'll be when you go to translate your model. Depending on the efficiency seen in this step, you may need to optimize your experiment. And this can mean changing the delivery method or trying new conditions, designing new guide RNAs, in fact, choosing a different nuclease. And we've spoken to many researchers who choose to move to higher quality or higher performing reagents at this step to increase the efficiency of their chosen modification. Using those high quality reagents will make creating your model more successful. This may mean several iterations of this, going back all the way, redesigning, choosing, and going through again. It's an iterative process, and you continue to make it more efficient as you move forward. Once you've settled on the most efficient reagents and have optimized, it's time to create your model. From there, do you need to isolate or select certain modified or edited cells? Do you need to perform whole genome sequencing? Lots can be done at this stage, again, to look at the entirety of the model. And then, once the model is created, you can be ready to move to the translation stage, where the focus is on scaling up the therapeutic, testing it in vivo, and demonstrating the safety of the model. For this first part of the talk, we're going to focus on the first two areas of this workflow, designing and evaluating efficiency. And we're going to talk about our discovery portfolio that has many different products for these two steps. While you can, in fact, use multiple formats for your guide RNA and nuclease, we're going to focus on two that we believe will be most efficient for downstream analysis. And that's the use of Cas nucleases in the purified protein format and synthetic guide RNAs. There are several advantages to the Rnp system, so that combination of a purified protein and a synthetic guide. First, it has very low cytotoxicity. Introducing these into most cell lines won't cause a toxic response. A classic example is using primary T cells. Introducing exogenous DNA invokes a cytotoxic response of the T cell. It's effectively doing its job. It's meant to get rid of exogenous DNA. So for CRISPR, that's not great if you want to modify primary T cells. The Rnp complex reduces that possibility. Rnps are also transient, which is great for therapeutic safety measures. Introducing a plasmid or even an mRNA that contains the CRISPR reagents, those reagents in the plasmid or mRNA form live much longer inside the cell line when they're introduced, and therefore the chances of them creating off-target edits or modifications goes up. With proteins and the Rnp complex, most are cleared within 48 to 72 hours following introduction, again, reducing that off-target editing, but causing the downside that we need high efficiency reagents in order to ensure high editing in that short window. Rnps then therefore have high on-target specificity because of their transience. They do work in non-dividing cell lines. They don't rely on the cell to express like they do in the plasmid format. So you can put them in non-dividing primary cells, again, a key model for therapeutic development. And they don't leave any genomic footprint. So they won't integrate into the endogenous genome. They're there, they're expressed, and they get degraded and move out. We have two different types of guide RNA are available. One is our two-part system and the one-part system. The two-part system involves a CRRNA, which contains that protospacer motif, about 20 nucleotides, matching the target sequence in your gene of interest, and a standard tracer RNA that would be the same for every experiment. The sgRNA, single guide RNA, combines the CRRNA and tracer into one, and there is some research that suggests these are more efficient than the two-part system. Both the sgRNA and the CRRNA, when made synthetically, can have stabilizing modifications added onto them to reduce the possibility of RNA degradation once introduced into the cell. So let's take a look at the many different options that we have for Cas9 proteins in our discovery portfolio. The first two that were published and discovered, of course, are our wild-type Cas9 and a new one that was created, a variant created shortly after the wild-type, which is called the nickase. So the wild-type Cas9 generally has very high gene editing activity. Many people use wild-type still today, despite new variants. The nickase, on the other hand, instead of making a double-strand break like the wild-type, it instead only nicks one of the two strands, therefore increasing its specificity. The downside with this is because it only nicks one strand, you actually need to have two sets of guide RNAs with nickases in order to utilize the repair pathway and get the indels or the integration that you want. However, it is more specific than the wild type. Another variant that we offer in the Rnp format that came shortly after the wild type is what's called the Dead Cas9. So this utilizes the nickase mutation and adds another one on top. This Cas9 can bind to the DNA, but it doesn't cut it. And this Cas9 is the basis for much of the CRISPR inhibition and CRISPR activation work that's done for regulating gene expression without causing those heritable mutations. One of the ones that we want to go a little bit more in depth into today is our Cas9 plus variant. And this has both high editing activity and on-target activity. And I'm going to show you how we developed that. So first, as I said, the wild type Cas9 has very high on-target activity. But as many researchers and we discovered through protein engineering of the wild type Cas9, there's a balance between the editing activity for the target gene and editing activity for off targets, or what we call specificity. Such that if you try to make the Cas9, this wild type Cas9, because it has very high activity on target, also tends to edit anything that's slightly related to your on target gene. 1, 2, 3 base pair mismatches often get edited as well. So the traditional wild type has very low specificity. From that came the enhanced specificity Cas9, where researchers undertook protein engineering efforts to increase the specificity. They found they could make mutations within the Cas9 protein that would reduce the off-target editing and therefore increase specificity. The unfortunate side effect is that it also reduced the on-target activity. So there's a balance here between on-target and off-target. And when you make changes to one, often the other is affected. So what we did through multiple rounds of protein engineering is look for a Cas9 that had both high on-target activity and high specificity. We tested both in parallel through the development of this variant, and we came up with a new variant, a novel variant that we call Cas9+. Here on this graph here, we have a look at the on-target activity, and we are comparing wild-type Cas9 in purple, the ESP Cas9 in blue, competitor Cas9 in green, and our Cas9+ here in yellow. And when we look at 3 different targets in HEK cells, we actually see that the Cas9+ maintains high levels of on-target activity. As you see here in blue, what I just told you on the previous slide, that ESP Cas9 had very low activity across the three sites. But what's great here with the on-target activity is when we look off-target, we look at a close off-target sequence that was differing by a single base pair. We actually see that the Cas9+ does not edit there. See here in purple how the wild type will edit at this off-target site at all 3 gene targets, but the Cas9+ does not. In fact, the Cas9+ has even lower off-target editing than a high-fidelity competitor variant as well. So it combines the high specificity of the ESP Cas9 with the high on-target activity of the wild type to make the most efficient Cas9+. From the discovery work that's done, again, the guide RNA design and choice is a crucial component of the CRISPR complex. And we offer standard single guide RNAs that can complex with the Cas9+ to perform this editing. Here, we're looking at a graph of multiple gene targets, looking at indel activity, comparing the standard sgRNA, so this is the 1-part sgRNA, to a competitor standard desalted sgRNA. And here, when complexed with Cas9+ and transfected into HEK cells, we see that the standard guide and Cas9+ have very high activity across the board at all sites, and in fact have significantly more activity than a competitor guide. Additionally, there was no loss of viability. So one of the key aspects of guide RNAs, because they are synthesized, is that they may carry with them artifacts from the synthesis reaction that could cause toxicity in your model of interest. And of course, as models get more sensitive, moving to primary cells, moving to animal models, that toxicity, associated toxicity with your CRISPR components, is a key variable that you need to assess when choosing your complex reagents. And we can show that our standard sgRNAs, in fact, offer very little toxicity and, in fact, are better than a competitor in this space. And therefore, the Cas9 plus with standard guide RNAs are a highly efficient editing complex for most model systems. Another tool that we have in our discovery bucket that can help in your workflow is our Cas9 Gfp. So this is really great because it allows visualization of gene editing. It's in fact a fantastic tool for an optimization of your delivery conditions. Often the addition of a GFP to a protein can have an effect on its function, but as you can see from the graph, Cas9 Gfp maintains very high activity, just like the wild-type Cas9 variant on which it's based. And it does so better than competitor versions that are attached to GFP. The image on the left shows how Cas9 Gfp can be used to measure delivery efficiency by measuring the number of GFP-positive cells. We work with several researchers, both internal and as collaborators, to use GFP to evaluate different formulations of complexes for delivery. In fact, one of the key trends in the CRISPR field is on nanoparticle, lipoprotein particles, different delivery methods for the CRISPR reagents for therapeutic uses. Cas9 Gfp is a great tool when you're trying to optimize a new delivery system or evaluate a new delivery system for your therapeutic, because it allows both editing and visualization in one Cas9. Once you have your reagents designed and you've started your experiments, one constant need throughout the workflow is to continually evaluate the amount of editing that is occurring. And there are multiple methods that differ in sensitivity, the time it takes to do them, and the cost to evaluate those editing outcomes. 3 of them are outlined here. The T71 digestion kit, which we'll go into a little bit more in a moment, it measures indel size and efficiency estimates. We recommend that it can be used for optimization of experimental design. For example, in those early phases where you're maybe designing 5, 6, 7 different guide RNA sequences to test out, this is, again, a great one to very quickly give you an answer of which guide RNA sequence is going to work and which ones need to be trashed. It doesn't require specialized equipment, no bioinformatic analysis. It has the same-day turnaround time, and it's very cost efficient. Comes in a single kit that you can do the entire experiment in one day. Sanger sequencing and next-generation sequencing provide much more detailed sequence information from the target site. Of course, these involve PCRing up your target site of interest, and then usually sending them out for reading on special instruments, right? And then the sequence reads will come back. So bioinformatic analysis is in fact required of those to determine how much of the sequence actually is cut and how much is not. It has a longer turnaround time and it tends to be more expensive. However, they will be more sensitive. Both Sanger and next-gen sequencing will give you more sensitive estimates of how much editing is done. So again, we recommend maybe to start with the T71 to weed out the guide RNAs that don't work. And as you're optimizing further down, you want to confirm that with these higher sensitive methods. A little bit further into the T71 assay kit that we offer, the first step here is to isolate your genomic DNA from your edited cells. You will PCR amplify your target region that has been edited. There's a denaturation step and then a reannealing. So what happens is these little pink spaces in here are presumably the edit of interest. And as they are denatured, they come apart into single-stranded pieces. And when reannealed, they don't always match up perfectly. And what you can get is mismatched duplexes. So on one strand will be a mutation or an edit, and on the other strand might be a wild type that didn't get edited. And those are the duplexes that are cleaved by the T7 endonuclease. Then you run those out on a gel, and what you'll see is a pattern of, in a negative control, you'll see no cutting because everything was wild type. And in something that has been edited, you should see a banding pattern underneath that should match up with where your expected mutation was. Here's an example, a typical example of the right here, where we have our transfection control that didn't have editing reagent, and then the edited cells themselves. You see there's still some amount that was not edited, and you can use imaging analysis to give a rough estimate of the ratio of these bands to get a rough efficiency number of how well your guide RNAs are doing. Again, this is a very fast, low-cost method for easy efficiency. So we've looked at these first two steps and a number of the discovery products that we have available to address them. Now let's look at this back end of the model. So as I mentioned, remember when I mentioned that the optimization often requires higher quality and higher performing reagents, and that many researchers switch to higher efficiency reagents at this stage. Well, we've innovated a new portfolio of products to address those needs and promote success in your translational work. Our recently launched translational grade CRISPR reagents are engineered to meet superior manufacturing standards that enable consistent and high quality performance every time. This new portfolio is called PURedit CRISPR products. While our discovery products are appropriate for most of your early discovery and even some of your preclinical studies, our PURedit products, in fact, have the highest performance, higher quality standards, and quality documentation that make them the appropriate choice and the best choice as you're moving your discovery work through preclinical and into clinical research phases. CRISPR's evolution has, of course, included a rapid move into the clinic, where CRISPR reagents are being used to generate cell and gene therapies to cure a number of human health issues and diseases. The attraction, or one attraction of CRISPR, is that the change is permanent. So that change is embedded in the DNA of the patient, which prevents long-term treatments or long-term drug use to control the symptoms. Additionally, cures for rare genetic diseases for which not much about the biological mechanism is known are now relatively easy with CRISPR, or at least easier than before. And as part of the democratization of creating new therapies, a number of new therapeutic companies have sprung up in the wake of CRISPR, who focus specifically on using CRISPR reagents for therapies. And the promise of CRISPR as a therapeutic was a major contributing reason why within 10 years of the first publication, the Nobel Prize was awarded to Jennifer Doudna and Emmanuelle Charpentier. So let's dig a little bit deeper into the PURedit portfolio and why it's perfect for translational or therapeutic research for CRISPRs. First, as we discussed earlier, the advantages of Rnps, these advantages play out for therapeutics as well. These are all key points: low cytotoxicity, transience, which leads to higher safety, working in therapeutically relevant cell model systems, and of course, no integration into the genome, leading to no long-term downstream effects. All of these are key points in why Rnps are quickly becoming the choice, the format of choice for therapeutic model development. So if we look at the two different pieces of this Rnp complex, we have a PURedit Cas9 protein in our portfolio, as well as a PURedit sgRNA. Our PURedit Cas9 protein has the highest activity of any Cas9 protein that we offer. It's also got the highest specificity of any of the proteins that we offer or that we've tested. It has 95% purity, and it's available off the shelf. Our PURedit guide RNAs, again, have highest activity and purity, similar to the protein. We include stabilizing modifications to prevent the degradation. These are made as custom products in 7 to 10 business days, and that's the quick delivery turnaround time for that as well. Combining the PURedit guide RNA with the PURedit Cas9 protein gives you the most efficient Cas9 complex for your translational work. So let's take a little bit more look at the data associated with these. So first, to look at the on-target activity. Here we're looking at PURedit Cas9 in red, two competitor high-fidelity Cas9s in yellow and blue. And you see here at 2 different gene sites in 2 different cell lines, the PURedit Cas9 is significantly higher on-target activity than 2 other high-fidelity variants. Additionally, when we compare the on-target activity to the off-target activity and plot that as a ratio, you see that the PURedit has the highest ratio. High ratio in this case is good. It means there's a lot of on-target activity with very little off-target activity. That ratio is sixfold higher than the nearest competitor. This is incredibly important for therapeutics. Because you do not want to have any off-target effects, any deleterious mutations as part of your therapeutic. That's a key safety parameter that's going to become more important and more strict as it's moving forward with CRISPR into the clinic. So when we dive into that specificity a little bit more, what we did here is here on the left, the letters are representing nucleotides in the protospacer motif, and in red are the nucleotides that we changed. And so off-target effects, of course, it means that that sequence, that 20 nucleotide sequence, is very similar to the 20 nucleotide sequence that you're trying to change. Sometimes they only differ by a single base pair, sometimes as many as 2, 3, 4 base pairs. As you increase the number of nucleotides different between your on target and a different sequence, the chances, the likelihood of editing at that off target site go down. With only a single base pair difference, there's a very high probability that this sequence would be edited to the same extent that the original target sequence was. So we made single base pair mutations across the length of the protospacer motif to look at how single nucleotide mismatches affected the specificity of the PURedit Cas9. And you can see here in yellow is the PURedit Cas9 and in purple is a high fidelity competitor. Resoundingly and across the board at 2 different sites and at every base pair we've tested, the PURedit Cas9 has significantly lower activity in most places, even in some places, even when it's only differing by a single base pair, the activity is almost non-existent. Again, this is a very important parameter for therapeutics, and so minimizing the chances that it would interact with any off-target site is key for moving things forward as a therapy. So not just the Cas9 is important for this very specific and highly efficient complex, but also the PURedit guides are important. So our PURedit single guide RNAs are purified by HPLC, which gets rid of and actively selects out any small nucleotides sequences that could be left over, as well as other trace amounts in the final product. Here, what we've done is compare our PURedit and our standard guides across 2 different cell lines, actually 3 different cell lines, HEK, T cells, and a mouse cell line at a number of different targets. And you can see at every target site, the PURedit guide RNAs have more activity than the standard guides. Again, suggesting that if high quality, high editing, and specificity is key to your work, the PURedit guide RNA is going to be the way to go, complexed with our PURedit Cas9 protein. So what we have here are 2 different tiers of portfolios with products to fit in every application, our discovery portfolio and our PURedit portfolio. Our discovery portfolio is going to focus on versatility, ease of use, pricing, and delivery time, of course. and it contains many of the Cas9 proteins that we've talked about, wild type, Cas9+, our Cas9 Gfp, and of course our standard guide RNAs. Our PURedit portfolio is focusing on high performance, highest quality standards, quality documentation, and is driven for therapeutic research. And there are 2 products in this portfolio as of now, PURedit Guides and PURedit Cas9. It's important to note that you don't only have to use PURedit products at the very late stages of your workflow. The earlier you bring in the high efficiency products, the faster your timeline will go. As I mentioned, as part of that workflow, often multiple rounds of optimization have to be done to get to the final complex before you create your model. PURedit Guides in Cas9 can cut down on that amount of optimization by giving you the highest efficiency early on in the process and reduce your overall time to the clinic or to your preclinical studies. So we're constantly adding and always working on adding new and innovative RNP products in both of these tiers to address the evolving needs of researchers at various stages in gene editing. And I'm going to highlight one of those ongoing initiatives for you next. So one of the next generation genome engineering tools that we're working on is what we call CRISPR-Chrome. And this involves a Cas9 variant that is fused to chromatin remodeling peptides. When we compare 3 different variants of fusions compared to the wild type Cas9, we look at indel rates. And first, here you see in gray is the wild type SP-Cas9 that has very low number of indels. So this could be these are gene targets that are generally hard to edit. Multiple rounds of gene editing have shown us that there are some gene targets that are just inherently more difficult to edit than others for various reasons. One reason could be that the chromatin architecture around them prevents accessibility to the CRISPR complexes. And so here you see all of these 4%, 4%, 3.8, and 2.2. These gene targets are very difficult to edit, and basically making any therapeutic that would target these or other targets that are difficult like them virtually impossible. How do you get to a therapeutic if you only get 2% editing? What we see here is that by fusing on these chromatin remodeling peptides, we actually increase the indel activity sometimes as much as 5 to 6-fold across the board for all of them. Having 25% of cells being edited is a much more workable cell number than only 4%, and this makes the therapeutic possible at this point. Additionally, we've looked at how these variants compare to the wild type Cas9 when we're trying to integrate a piece of DNA. So we have very low integration here, 6.5% with the wild type, but these variants show increase as much as 5 to almost 6-fold here across all of them. Again, taking that efficiency higher to allow this therapeutic strategy to be successful where it would not have been in the first place. And this is just one example of new technology and innovations for our Rnps that we're working on. So what's next for CRISPR and CRISPR therapeutics? So to date, many of the therapeutics and the kind of the headlines that I showed you have largely focused on easier targets, the low-hanging fruit, if you will. But there's a number of different areas where therapeutics are moving in kind of that next generation genome engineering, including inserting DNA to repair or replace harmful DNA sequences, multiplex editing, so multiple genes being edited at one time. You can imagine that this increases efficiency while decreasing safety concerns. If you can edit 2 to 3 genes at once, you don't have to go back and do them sequentially. A lot less time for off-target edits to occur. Base editing and prime editing are 2 new forms of what have been termed precision editing, where they're using CRISPR components to directly change bases without making double-strand breaks. In the base editing, you can directly change single DNA bases, and in prime editing, you can change both single bases and longer stretches of DNA. The advantage here is that without the double-strand breaks, there's less impact to the remainder of the genome. It doesn't create breaks everywhere else in the genome and have that off target. So again, safer and more precise. And of course, then CRISPR inhibition and CRISPR activation, the ability to turn genes on and off without making breaks at all, is a key area of therapeutic research as well. So what do we need? What does CRISPR need to be successful in translation? What do we need to continue to work on to get it to this stage? First, better specificity without decreasing efficiency. And I showed you one variant that we have, well, two variants, the Cas9 Plus and the PURedit Cas9, in which we have increased specificity without decreasing that efficiency. Variants that have this are going to be key candidates to move forward. More precise editing, editing single nucleotides, inefficient precision editing can limit the field to therapies where gene functions need to be eliminated or where corrected cells gain a competitive advantage over the unrepaired cells. Additionally, improved access to target sites. So right now, we are limited to targets that have PAM proximal regions, as well as to sites that can be efficiently edited. So inclusion of variants to the nucleases that expand our accessibility to the rest of the genome, virtually making it able for a researcher to edit anywhere that they want without limitation will be key at moving forward. And finally, delivery. Delivery is another key factor here. Whether the CRISPR reagents are being used to modify a cell line and the cell line becomes the therapeutic introduced into the patient, or the CRISPR reagents themselves are the therapeutic introduced into the patient, the delivery of that can have a huge impact on the efficiency of it as a therapeutic, but also on the safety of it. And so continuing to innovate in new methods of delivery, new, safer methods of delivery is going to be key to continue to pushing CRISPR forward. So as I've shown you throughout this webinar, we provide a number of solutions that can cover the end-to-end workflows for bench to bedside using gene editing. Thank you for your attention to this, and I will take any questions.
Thank you, Jennifer, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Is PURedit Cas9 different from Cas9 Plus?
That's a great question. And so the base Cas9 variant, the mutations in Cas9 that make it more specific and highly active, are the same for Cas9 Plus and PURedit Cas9. The difference between the two is the PURedit Cas9 has an enhancer in it that raises that level of activity even higher over the base Cas9 Plus. And so that's present only in the PURedit Cas9 and potentially in future PURedit Cas9 products as well.
All right, thank you. Next question. Does the T7E1 assay kit contain the DNA polymerase and the master mix to perform the PCR?
Yeah, that's a good question. So the kit does contain the master mix. It does not contain the DNA polymerase. And the reason, one of the biggest reasons for this is that the PCR conditions, including the polymerase, may already be worked out in your own lab. You may already be using a DNA polymerase and have optimized PCR conditions for your gene interest that you would then like to use to amplify that DNA to do the T71. So this way you can use a pre-optimized protocol. If you don't have one or you would like to buy a polymerase from us, we've actually worked out with the kit how to use and get the best results with our Accutech DNA polymerase. And that data can be found in our T71 page and is also linked on the T71 product page, so you can buy them both at the same time.
Great, thank you. Next question. This has kind of two parts to it. So looking forward to future uses of PURedit, um, will the Cas9 be able to be conjugated to GFP? And will the CRISPR portfolio utilize primer or base editing?
Yes, so this is very forward-looking, right? Of course, this portfolio for PURedit is new, just launched this year. And as we move toward more precision gene editing, I think we're all looking forward to base editing and prime editing. The GFP conjugation itself makes Cas9 a good molecule for tracking that transfection efficiency. So I think these are all options for the PURedit portfolio in the future. And I would encourage anybody who wants to be kept aware of these options to sign up on our website. And so we'll be sending out information to users and customers on a regular basis to keep them updated on the newest products that we have in this portfolio.
Thank you. Next question. Do you have a library of gene knockouts such as RNA binding protein gene KO library?
Yes, we do have several options for libraries for gene knockout. We also have several options for shRNA binding libraries. These are all present on our website. We utilize many of the known knockout libraries that have been published, and you can find all of those. We have very large, extensive collections. You can buy the entire library. You could buy a subset of the library. You can buy pools of the library. Actually, the options for your custom-built library are pretty endless with us right now. I would encourage you to get in touch with your sales representative or come online through our customer service, and we can get somebody helping you build that library.
Great, thank you. Next question. How do you design gRNA for prokaryotes, and how would you validate it?
So that's a little different of an approach from this. Everything that we've talked about in this webinar is for a mammalian system, right? And so something very interesting is that even though CRISPR was found to naturally exist in bacteria, CRISPR and Cas9 in its normal state don't actually affect gene editing in prokaryotes and in bacteria. So we have devised a new system, and you can, again, find this on our website. It's our bacterial gene editors. It's a Cas9 fused to a traditional bacteria editing system. It's plasmids designed specifically for work in E. coli. Most of these that you're going to find are not going to be good for work in E. coli because they don't contain any of the promoters and things necessary for expression in bacteria. So I'd highly advise you to go to our website, look for our Cas9 BAC1P products, which do work specifically in E. coli.
Great, thank you. Next question. What is the best delivery method for the PURedit CRISPR product?
So we've tested a number of delivery methods, and it's going to depend on the model system that you used. We recommend electroporation, and many of our results are done with electroporation in the immortalized and primary cell lines because we find that's what gets the highest efficiency, and that, in fact, for the PURedit, that enhancer also has the highest efficiency when done with electroporation. However, we've also tested it using liposuction kits and had users test it with like microinjection into embryonic models. And both of those still show same high efficiency. So it really can be optimized to whatever is necessary for your model of choice.
Thank you. Next question. Can I use PURedit Cas9 with standard sgRNAs or PURedit sgRNAs with a different Cas9 variant?
Yes, you can. And so there's no magic formula between the PURedit guide and the PURedit Cas9 that makes them only work with each other. Of course, the best and most efficient CRISPR complex is going to be a PURedit guide with a PURedit Cas9. However, we do know that people are working on their own Cas9 variants. Maybe in your lab, you've done your own protein engineering and you have a Cas9 that you really want to work with, but you're looking for a high efficiency guide guide RNA. In that case, you could easily pair our PURedit guides with whatever Cas9 variant you've already developed. And so, yes, the obvious answer is that they are not exclusive to each other. They can be used with other variants.
Great, thank you. Next question. For translation to clinic, how important would it be to have GMP grade guide RNA for complexing with GMP Cas9 to form Rnps?
Yeah, so this is a great question, and it underlies what you're going to actually be using the CRISPR components for. So if you're using the CRISPR components, let's say, as the therapeutic, the thing that you would directly inject into a patient, then in that case, GMP is very important once you get to that clinical level because it's obviously going directly into the patient. However, if you are going to use CRISPR to modify a cell line, and then that cell line is in fact the therapy that's going into the patient, the CRISPR components should themselves be of the highest quality, but do not have to be GMP grade, as according to the regulations from the regulatory bodies in Europe and in North America. That is true right now. And so as you translate that cell therapy into the clinic, you still need high quality CRISPR reagents, but they don't have to be GMP. And that's why this product is so good. It gives you that high quality with that high activity in the PURedit, but you're not waiting the long custom times or paying the price for the GMP. That's unnecessary for those early clinical stages.
Great, thank you. And it looks like we have time for one more question. Can I use PURedit CRISPR reagents even if I'm not developing a therapeutic?
Yes. Of course, we've talked a lot about therapeutics as part of this webinar, but that doesn't mean that if you're creating a model to better understand a biological pathway, that you can't use the PURedit reagents. PURedit is designed to be highly efficient, so high specificity and high activity. And we recognize that there's a place in early discovery where that high specificity is not always required. However, if that is important for your model, if that's important for your research, then of course you should be using the best reagents for your experiment, and the PURedit will do that. I mean, it'll give you that high specificity and high activity right off the bat. And so no, if you are not relegated to our discovery portfolio if you're not doing a therapeutic. You should feel free to choose anything across either portfolio and mix and match between the portfolios if necessary.
Well, thank you again, Jennifer, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Millipore Sigma, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.